So as per the NCERT, it clearly says if you try to insert a piece of DNA into any host cell, it will not survive, it will not multiply, it will not pass on to the next generation. But yes, if we successfully manage to join it with the genetic material of the host organism, then every time this host organism is going to divide, or this host organism is going to show replication of its DNA during S phase of the cell cycle, then every time it replicates, even the alien piece of DNA will replicate, and now the next generations, all the cells produced in the next generations are going to have the same alien piece of DNA transferred because of the sexual reproduction, or otherwise the asexual reproduction. So this is the main concept of the biotechnology. Why this replication has occurred? Because there is always on a chromosome or on a genome, or even the plasmid also, there is always the origin of replication. So we have already learned in the genetics unit that origin of replication is the place from where the replication starts. So if we successfully insert an alien piece of DNA into the genome or into the chromosome of an organism, and because that chromosome or genome carries the origin of replication, so the main point here is necessity of having origin of replication. If this origin of replication is not there, then the multiple copy making will not take place, and it will never be successful to transfer it to the next generation. So origin of replication is responsible for initiating replication. Therefore, for making multiple copies of an alien piece of DNA in an organism, it has to become a part of chromosome, and that chromosome should carry the origin of replication. Thus, an alien DNA linked with the origin of replication is able to replicate and multiply successfully inside the host organism, and this is called as cloning, or making multiple identical copies of any alien piece of DNA. I hope this point, I have read the last lines from the NCERT itself. Now, first time the construction of an artificial recombinant DNA molecule. Now let's understand who performed this experiment. So the concept of development of principle of genetic engineering, it revolves around two things which I already discussed. One is what is plasmid in bacteria, and we all know plasmid is the extra chromosomal circular DNA. We have learned this thing in the 11th class also in detail. So plasmid is able to undergo replication along with as well as independent of the chromosomal DNA. So whenever a bacteria thinks of dividing, it will first of all replicates its chromosomal DNA, or what you call as prochromosome, because the chromosome word is basically used for the eukaryotic organism. So prochromosome. So whenever the chromosomal material of the bacteria divides, then there is something else present inside the bacteria, and that is the extra chromosomal DNA, and that DNA is circular in nature and provides special properties to the bacteria, and that is called as the plasmid. So the discovery of plasmid and how it functions and how does it replicate, and then the presence of restriction or discovery of restriction endonuclease enzymes, which breaks the DNA at a specific site, or what you also called as the biological or molecular scissors. These were the main thing which gave birth to the genetic engineering. Now Paul Berg in 1972, who is considered as the father of genetic engineering, successfully introduced a gene from SV40. So the virus is SV40. So from this virus SV40, he took a gene out and introduced into the bacteria with the help of a virus which is called as the lambda-phage. Lambda-phage is a bacteriophage. We all know bacteriophages are those viruses which attack, or for them they are the parasites, for them the host is bacteria. So the technique of the genetic engineering includes formation of a recombinant DNA. Recombinant means when two different sources of DNA are joined together, you call it as recombinant DNA, rDNA. Now this is done with the help of molecular scissor and molecular glue. So once the recombinant DNA is made, how we made? We took the alien DNA source, and then we take the vector. What is vector? For example, plasmid. So first of all, we make two cuts into the source DNA so that we can take out, imagine a long piece of DNA is there and you are interested in this much gene, so definitely you may be making one cut or otherwise two cuts. So the maximum time two cuts are made, and this piece of DNA is the DNA you are interested in. Now you take it out and join it to the plasmid. Now plasmid is circular in nature, so you need to make only one cut and then insert this piece of DNA in that cut. So with the help of scissor and with the help of glue, because glue is going to join them together, molecular glue, DNA ligase. So the use of the gene cloning. Now how to make gene cloning? That you insert it into the organism, or otherwise there are new methods like PCR, we'll be talking about later in this chapter. So we can make multiple copies of the gene you are interested in and then transfer this gene into the target organism. Target organism is the host organism, and remember, in this concept we have only transferred the gene of interest. Gene of interest is mentioned in biotechnology as GOI, or the desirable gene. So this is the conceptual development of the principle of the genetic engineering, and this was for the first time explained in detail by Stanley Cohen and Herbert Boyd in 1972, because they successfully transferred a gene for antibiotic resistance. They took out this gene from the plasmid of Salmonella typhimurium. Remember, it causes typhoid. Typhimurium causes the food poisoning. Salmonellosis is the food poisoning caused by it. So Salmonella typhimurium's plasmids, a particular gene was taken out and then it was joined to the plasmid of E. coli, and that plasmid was inserted back to the E. coli. Now E. coli has become genetically modified. We will continue in the next class from this experiment.